So let's look
abnormal.
at the class we want to pair first, because I know we talked about pairing second period together, because I have some that really need some extra intervention. And I know you have some great kids in there that are some high flyers and really just understand everything. So I thought it'd be good, since we've combined them before, to just combine them again.
Good. And they've got buddies in each class that they work well.
Yeah, like Jake and Jake work really well together. So, and I think, like, especially with yours was question number 5, my largest discrepancy was question number 5. And I think what they missed on that is they did not put together that heterozygous type B is BO.
Yes.
I think that's what they struggled with.
They would have gotten these two, but would have not been able to get those two.
Exactly. And a lot of mine did not click
and
BO.
that's the same trend.
Yep. So I think they understand the basic blood typing, but I think they struggle still with just how to write the heterozygous when it comes to blood typing because of those multiple alleles.
Because it does that separate, it does it differently. Yeah, because all of mine who missed that, with the exception of one so far, it's the same thing. They made the same exact mistake.
And even with question number 1 with being vocabulary, they would only mix 2 of the questions up. And one of my biggest ones that they mixed up was dihybrid and codominant, because one said 2 traits and one said both, and they knew that di meant 2.
Okay, so mine are a little bit more all over the board. But I also have a very large range in reading skills in this group. And this was the most wordy one.
And I mean, it is 7 questions, so it's a lot.
Same mix, and I would have expected her to have gotten them all.
And I feel like a lot of them did not use key terms, like for polygenic or polygenetic. A lot of them did not pick out the many. They just automatically went to more than 2, even though the more than 2 said more than 2 alleles. Yep.
And but that's something that we harp with them. This keyword identification. We do that all the time. And this is why.
But even our review yesterday, the review we did yesterday, we literally had them do a vocabulary term, practice that. Vocabulary term, practice that. Vocabulary term, practice. So I actually talked to a couple of my groups today about how to actually study, because vocabulary is one of those, you either know it or you don't. You studied it or you didn't. Vocabulary is not applying. Vocabulary is just basic recall. Now, the other questions we had them do, those are application questions. But this one, I kind of got onto mine a little bit because they're just not reviewing their notes like they should.
And, I mean, realistically, of this particular class, I may have one kid who goes home and does stuff outside of class.
In this class, I maybe have 2.
So,
like Ella and Skylar, they're like my...
and
they're like my top kids.
Kerrigan's that way for me. Like, you could tell the very next day they come in and they know their vocabulary.
Yes.
And they do that because they went home and they studied. So knowing that realistically they're not studying, we've got to build in
that study time.
The repetition,
yes,
practicing that keyword association so that they can build that skill.
Yeah. I think for test review on Monday, I think we need to have like a vocab station.
Okay.
Just where... Because if they know these vocabulary terms, they are better inclined to understand the special types of Punnett squares.
That's very true. And maybe we could do one of the vocabulary activities that we're pitching for the literacy team is a keyword. To give them the term, and then you give them the keyword, and then the full definition.
Yeah.
So they match just the keyword first, polygenic, many genes, and then they have to find it within the context of the big, messy, fancy science sentence.
Well, and one of my honor students today, he actually said, Latin really helps you with this. I said, welcome to science. They heard Latin-based words.
We should go back to teaching Latin. All right. Were there any other questions that just were off the charts kind of?
I didn't have a ton that were just straight missed. So I don't think there were any questions that we really need to tweak or kill completely. I think it was just like that one right there. They struggled with reading the sex-linked Punnett square. Even your largest red one, number 4, that was a vocabulary question. It was the mini genes, and they just did not choose polygenic. So that is not a bad question. It's just they didn't know that vocabulary term. And you can see that back in question number 1 as well.
But I had more kids do better on this than on number 1.
I did too.
But I think it's the example. Like having the example and remembering, I remember we specifically discussed this, as opposed to two phrasings or descriptions that are really close. All right, so if we are not displeased with the quality or the statistics of the questions, and if we feel that what is missed is an indication of not an error in the question, but understanding of student performance, are we good to start pairing our kids?
I think so. And I think us doing the pre-quiz at the beginning of class, that helped me because I kind of did a mini version of this during class today. So I had all the kids, I'm like, if you made below 50, I'm just going to call you out. Go over here because these grades don't count. If you made an A or a B, come over here. And I literally paired them together. And that worked really well.
See, as they came in, I was kind of watching and going, oh, so-and-so's getting vocabulary-based questions right, and so-and-so's getting Punnett script quiz. I'm going to put them together. And so a lot of times, like, they couldn't pick out the smart kid in the pair. And then they were more, like, sometimes they just get tired of being the helper.
Yes.
And sometimes they get tired of being the one who always has to have a peer tutor.
And when we assigned our practice problems that we decided on, I actually assigned it to the lower scoring student. And I'm like, you're the one that's going to come up here and teach this. And they're like, I'm like, you can do it.
Yeah. All right. So we saw that the pairing helped because we saw drastic increases in the pre-quiz and the real quiz.
In this class period, I went from a 51 to a 73 average. Now I know they weren't the same questions, but they were all the same concepts.
They're quite comparable in difficulty level. And, you know, if you can do one codominant Punnett square, you can do a codominant Punnett square. Okay, so I sorted mine based upon like, so we have our document here where we've got a learning target, all the learning targets that were on this quiz. And while that's not everything for the whole test, it's what we're focusing on from this particular quiz. So we had 3 learning targets, and one kind of has subparts to it. And so we split them up by matching them per a question on the quiz, and then we have indicated which kid missed which learning target. Okay. So I have a stack of my kids that, like, they need all the help they can get. Sure, they got one Punnett square right, but they can do 101 and it's not going to hurt them. Then I have my high flyers who I honestly think whatever Punnett square you give them, they're going to figure it out.
Exactly.
And then I have my in the middle. Like, it seems to be dependent upon, you know, they're good on these types of Punnett squares, but they really struggled with sex linked or blood type. So do we want to put our kids who need all the help they can get with our kids who can give all the help they need?
I think so, because I feel like that's going to help our students that have mastered and shown proficiency, more than proficiency in everything, a chance to actually do something.
Yeah.
So I think that would be good. Okay.
Intermediate.
So then, remember what we were talking about is as we do our stations, we're going to have like the versus stations. We'll have sexual versus asexual. We'll have Punnets and all that. But then up here, we were going to have an intervention station with us. So basically, the pairs will be split again when they get up here with us.
Okay, so they're going to travel through test review with their person.
With their person.
And their person is going to help them the whole time.
Yes.
And they're going to go together. And like, I know that this was just for these 3 learning targets, but typically you see the trend.
Yes.
If they could do these hard Punnett squares, they did fine on the regular Punnett squares, so that'll be fine. Okay, so
I think up here, you know how we like to use the whiteboards to see what they're doing. I really think up here we have some sample problems.
Okay.
We let them do whiteboards so they can show us, because we can be up here with them.
For our kids that we flagged as intervention kids.
And with us using both rooms, you know, it doesn't matter which kids we get. We'll just be like, okay, let me see your paper. Okay, extension, you're going to go over here. These are your options for this. But then those intervention kids, we take them to try to grab them one more time to preemptively get them before the test on Tuesday.
Got it. So we're going to take our kids, okay? We're just going to split them, half of them in your room, half from my room, because most of their partners are cross-class, okay? They're going to go through the stations.
And we can be more intentional with our station, because remember, my student teacher, she's helping set up the stations as well.
Oh, that's right. So you'll have an extra person. What period?
We will have her in second period.
Second period.
We will have her in second period.
First, I have Gerard for inclusion, and then third I have Roland. So we're going to have an extra person in every single period. So a lot of that,
they can help with that
on task management.
On task while we are actually providing
intervention.
Yes.
Okay. All right, so we've picked all of our review activities, okay, and we can finish tweaking those because we've got till Monday before we actually have to do those.
And Friday, we can do.
PD on
I
Friday.
also asked Annie if she wants to come. She's more than welcome to
Awesome.
plan with us. So
Okay.
yes.
So we can tweak those activities. So by pairing the kids, hopefully strategically,
I hope so.
you're hopefully going to have cohesive working partners helping each other, strengths and weaknesses, trying to balancing out and taking into consideration social personalities. And then when they get to us, we're going to take the kids who haven't learned, who haven't hit their learning target, and we're going to, after they've worked those other stations, we're going to be checking with them again.
And I think up here we really need to focus on meiosis, letting them see those phases again.
And these complex Punnett squares.
Punnett squares,
especially the multiple alleles and sexing.
Yes.
All right, so then the kids so you brought something.
Yes, I did.
So,
And we may even give them options.
they could choose, absolutely.
So, we've used these genetic cards before.
Is this where our ethics came from?
Yes.
Okay.
But there's still some in here that we haven't used. There's crossing traits. We have traits here. We have more crossing traits. We have genetic disorders, human reproduction.
Don't like genetic disorders.
Yes.
Now,
So
are
here's some more
these
genetic
all-
disorders.
Okay, so these are more Punnett squares.
Yes.
So we could even keep some of these over here with
We could.
us as the kids who haven't learned.
So disorders, but
Genetic
then you get
disorders,
into
right.
genetic engineering and cloning,
Okay.
and then dog breeding. So we talked
love
about,
dog
I know. So we talked about, choosing traits.
Mm-hmm.
I don't necessarily think the human genome, but look about this one, twins. They have all had questions about twins because we have like 8,000 sets of twins in this freshman class.
Okay, so these could be fun. So the other thing that I found was mutations, because mutations is in here and we didn't really have
Like a lot of activities with it.
a lot of mutation activities. And so what it does was, no, not this one. There we go. Yeah. So it gives like examples of mutations that I don't know, are not overly
See,
weird.
I've been telling them since I'm a ginger, I'm mutated. They
And
didn't
I
believe
agree
me.
But what it does is I think it introduces, so it walks through the percentages, it walks through the actual names of the protein.
Oh, I like that.
Because we don't ever go into that level of detail.
Yeah, we don't ever name the genes.
Yeah, because they're always just random letters. But our kids who are that high are probably going to be some of your AP kids, or the ones who are
Yes,
going to be
that are going to need to see those types of terminology.
And then what it does is it gives them examples in other species.
Oh.
And then they have to, that it clarifies if it's just a variation disease or both. And I think our standards harp on the source of variation.
Yes.
But I think really hounding that will go well with the next unit of natural selection.
I like
with
that.
Variations being part, and then like if it's because it will give you like a negative, like if it's both. Sure, like your red hair, yes, it's a trait, it's a variation.
Yeah.
But then you've got higher association risks of skin cancer and things like that. So then we can start getting into this concept of per environment.
So what if we made some type of handout or something that kind of guides them through this site? Maybe even picking 3 that look the most interesting to you.
Okay.
And then we choose a few of these genetics cards, and this be our extension and flex next week. So if you will make a handout for our extension station for test review, I will go through these cards and I will pick a couple that kind of mirror our concepts already that we have for our review stations. And then I will go through and I'm going to pick a couple for flex next week. And then I'm going to pick a couple for after the PSAT.
Okay. I'm going to do this action plan in our email.
Because a few of these kind of will preface evolution as well. And those changes over time. Yes. I think it's a great segue into it. Now, how about we try to have these things done by Friday?
Because then we can look at it.
So that way we can go ahead and have everything set up and everything good to go on Monday.
Okay. Review stations. I'm doing the review station for the mutation things, and you're handling the Let's Talk Genetics cards. And then we will finish tweaking the other final stations with
Yes.
student teacher as well. Okay. All right. And then, okay, so we've looked at our learning targets. So our focus today was on these particular 3 learning targets with
Yes.
complex Punnett squares. We have strategically paired our students to help the kids who are not learning.
Yes.
And then we're going to additionally do small group intervention with them as a station. Our kids who are learning well, they're the ones that are on that last split station, are going to go do the mutations thing.
Yes.
That's the kind of interesting but more complicated concepts. So I think with there being 2 different standards in this unit, it might be good to decide if you need to master. Oh, that's the, we got the
Yes.
email. If they need to master just the meiosis standard, just the
Yes.
Punnett square standard, or both. So we can
Yes.
kind of split it up that way.
I think that's perfect.
And we'll put that in our action plan.
Okay, thank you. Thank you. Would you also print out one of our templates for mastery real quick?
Yes.
Perfect.
On Friday, figure out mastery for 5. Okay. And then the other thing is the test. So I went through and I pulled some questions from last year's test and I made sure that we had each learning target covered
Perfect.
in a question. And then we may want to, that's something we can look at on Friday. Yeah.
Yes.
And then I can run it with the other bio teachers today too and get
Oh,
their,
perfect.
I can,
Okay, good.
I can get them to look at it with me today while you're in Guiding Coalition, and that way we can get feedback from the upperclassmen
Absolutely.
bio
Perfect.
teachers, and then we can add cumulative stuff. Okay.
And
So
I
we will,
and I think we should do just like we did for the last test, make it worth like half a point.
for the cumulative stuff,
Yes.
so we can get a better, the score's a better indication of learning targets
This
on
unit.
this particular
Yes.
unit.